Okay, today we are going to explore the fine world of Luminex data analysis. I think you've already had a little talk about how Luminex technology works, right? That we have the ability to use these beads to let us know about the presence of things that are present at very, very small levels. It's like an immunoassay except on multiplexed to a very great extent. But there are lots of things that come into play when we try to make sense of these data. And frequently they are highly problematic to deal with from a statistical point of view. Today I want to try to walk through some of these key concepts and explain why they can cause particular problems. Specifically, we're going to be talking about calibration curves because we use part of every plate to establish that calibration curve. Why are we doing that with every plate? We're going to talk about missingness, which is the situation that comes about when you expect to have a number for a given analyte in a given sample, and that number is missing for some reason. And we're also going to talk about normalization, which is kind of the problem of how we deal with data when they do not have a nice bell-shaped curve to them. Okay. So we're going to start with several key terms. I don't know yet what the assessment for this unit will look like. Maybe you're going to ask me for questions to go on a paper of some sort, or maybe this is not part of the assessment, I don't know. Okay, well, it probably will be. So I would just note that typically the questions I ask in assessment are about vocabulary, and you should be able to distinguish a plausible sounding but wrong definition from the true definition for each term. That is definitely my favorite kind of question. So I tend to write devilishly real-sounding definitions for things that are fake. So just be forewarned. If you ever catch me using the term reverse translation in a test question, you know that that's false, right? Reverse translation doesn't happen so far. Okay, so I would just try to introduce some of the vocabulary terms that I want everybody to be able to take away right here from the outset. So we're going to talk about calibration, level of detection, and level of quantitation. LOD and LOQ are those last two. Specifically, given some fluorescent intensity that's come back from the Luminex box, how do we interpret that as a quantity? Next, we're going to talk about missingness and imputation. So missingness is the problem, imputation is how we attempt to solve it. This is what happens when we have missing measurements within our experiments. And finally, we're going to talk about normalization. How can we minimize the impact of the most extreme measurements that we see in an experiment? And although I'm talking about these in the context of Luminex, they apply pretty much across the board. There's a world of quantitative experiments out there and quantitative technologies out there. All of them struggle to some extent with missingness. All of them need the background of calibration for us to be able to turn a measurement into a quantity. So these concepts, although they're being introduced in this context, can be used much more broadly than that. Okay, so why bother with calibration at all? We are using real estate on our plates to do this calibration every time. And if we're going to do that, we need to be getting some sort of value out of the calibration process. So we'll try to talk about that. So we start with the fact that biologists are really most interested in concentrations. They don't really care about what the fluorescence is. Their goal in the end is to be able to quantify something. So if you can't translate this amount of light into a quantity, a lot of times that information just seems like you have a failed experiment, essentially. So being able to deal with that is the first thing. Next, we frequently have far too many samples to fit them on one plate. Right? If that's the case, then we need to be able to evaluate a concentration, well, we need to be able to translate a fluorescent concentration from this plate into a concentration that we can compare to on another plate. If these samples are to be compared, they need to be put on the same footing. That's what the calibration is intended to do. We need to know the variability of measurement with respect to concentration. Now, a lot of people, I think, get the calibration series is intended to say, with this much light, you have this much quantity, right? But we would also like to have a model, if you will, on how much variation there is between these measurements and the quantities they're supposed to represent. If the calibration curve samples are all right on the line, then we have a low variability of measurement. If the curve that we fit through the data is a pretty loosey-goosey fit to the data that we've got. If there's a lot of variability there, then we have to take every concentration with a big grain of salt. So being able to evaluate both of those axes is important. It's a little weird having this with the slides completely behind me, but I'll try to keep my voice this direction. Okay, so in a typical strategy, we're going to give over some number of wells. Now in this case, I've specified that we're going to use 16 of the 96 wells of a plate for calibration. This is not how it's always done, and different sites are going to have different approaches to this. I mean, you could imagine doing Luminex off something other than 96 well plates for that matter. But in this case, we're setting aside 16 wells of a 96 well plate. You can see that we've got a concentration series shown in kind of an exponential series at the left. That we have a very large bar at the left that's showing the maximum amount that we spike into one of the wells, and that correspondingly should get the most intensity. But then you can see that what we have is a very rapidly descending exponential series, that each well after that gets half as much sample as the one before it. So we started with x spiked in, and now we have x over 2, x over 4, x over 8, x over 16, and so on. So that shows a really rapid decline in the linear amount of sample that you've got. Now I'm showing the same data a second time on the right. And this time, instead of showing the linear concentration, I'm showing it on log scale, specifically log base 2 scale. Now that's valuable because in the first case, we decided we were going to spike half as much in each time. And by showing it on a log base 2 scale, it means that we drop by 1 unit every time. That's kind of the magic here, that we're dividing by 2 on a log scale. That means they're evenly spaced one apart. So you could ask, why not go for a bigger cut down? What if you were to do 1/4 as much from each bar to the next, from each sample vial, calibration vial to the next? If you were to do that, you would have a much bigger difference in intensities between successive wells, and you might find that your ability to pin down where exactly the bottom of detection is would recede for that, right? So if you were to do these 16 wells a number of different ways, you could get different kinds of information. If you were to do, let's say, the first 4 vials are all the same concentration, the next 4 vials are all 4 times less, the next 4 vials are another 4-fold less, you would have a better idea about variability at those points, but the total geometric range over which you were getting calibration information would drop quite a lot. Right, so when you invest your 16 wells into this calibration curve, you're making an engineering decision, essentially, about how wide a range you're going to have calibration information for, and by how much in discerning how much variability you're going to detect at any one concentration. All doing okay so far? Okay, great. So this is the other stuff. I wanted to mention also that we're not just calibrating with known spike-ins, we're also calibrating with blank samples. And that's going to be really important when we come to the place where we talk about level of detection in particular, limit of detection. Okay, so now we need to construct a curve that shows how the known amounts that were spiked into these wells relate to the observed intensity. You can see here that we're, again, showing the median fluorescence on a log scale, and we're showing the log concentrations on this x-axis. So we have a known amount that we've spiked into these vials, we have a observed amount of intensity, and therefore we can say, when we have this much present, we expect this intensity of light to respond to it. I want to note, though, that we have a couple choices that have to come into play. One is that we're going to have to fit some sort of function to relate the intensities to the known concentrations. So typically, you'll see people using things like a logistic regression. A logistic regression assumes that you have some basal level that reflects the kind of signal you get when there's no there's nothing spiked in at all, and you have some maximum amount of intensity that results when you have some very large amount spiked in. There are a couple oddities about this, so I want to remind you of them, even though I think you probably already know them. One is that you're going to get some amount of fluorescent intensity even when nothing at all is present in the sample. Okay, so this background fluorescence is something that we're always having to fight against. If you're trying to make a very accurate measurement of quantity when you have a vanishingly small amount of analyte, you're always going to be fighting against this background light that you can't really get rid of altogether. The other thing is that when you get all the way up to the top of that concentration range, there's some amount of muting effect. I don't know if you've all had this experience, but today I had a person drive up behind me at a traffic light, and he had a very loud stereo going on. He did not, however, have a really great set of speakers, right? So he had his music cranked all the way up, and that poor speaker was doing the best it could, but it mostly sounded like bah, 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 right? So that's the same effect that you get out of a Luminex box. When you get to the very top of concentration, there's just a limit on what the maximum amount of light can be read off of this well is. And as a result, there's some maximum past which you can't push and get a sensible concentration measurement. We have some light attenuation at the bottom that's causing signal when nothing is there, and you have some amount of attenuation at the top where more sample does not lead to more light. Okay, so the other thing I want to note is that we have these kind of dotted lines here that reflect a variance of our model, that the points don't all fall perfectly on this logistic regression fit. They fall close. We did the best we could with this logistic function, but there's still some amount of variation in the vertical positions of observed intensity and what we modeled to be there after fitting all the points. The less, the more noise you have, the greater the distance between that curve and the calibration fit, the worse fit is that calibration. The software is going to do the best it can to give you a calibration curve. It is not, however, guaranteeing that that calibration curve is going to mean anything. You should, on every plate, check that the calibration curve actually overlays the points to which it was fit. If you see, for example, that you put a greater concentration of analyte in, but the intensity fell, you're in a world of hurt, right? So we want to be cognizant of the fact that these models are only as good as they're fit to the calibration points. Okay, so now I mentioned as well that we are going to shoot some samples in which no sample was spiked at all. That represents our way of evaluating the level of detection. If you have no sample there, but you do detect intensity, that is some indication of where this background radiation line falls, that we have some basal level of intensity that is going to show up whether or not we have sample there. And that's what this green line is attempting to show. Okay. So we've made passing mention now of LOD. We've talked about this logistic regression to fit levels of intensity versus known concentrations. Next, we need to talk about limits of quantitation. Limits of quantitation are, I think, kind of poorly understood, frankly. And part of this is the idea that people think, so long as you have enough sample, you're within the limits of quantitation. But it's not one limit of quantitation, it's two limits of quantitation. We have a value below which we can't detect, and a value above which we can't quantify, right? So the quantification limits are asking where is the variance of this model low enough that we can trust a quantitative value derived from that fluorescent intensity. So one way to think about it then is to ask, what is the coefficient of variation that we see? Now, I think you guys haven't had the biostatistics unit quite yet, and I'm sure you're going to enjoy it thoroughly. Everyone always does year after year. But we should have just a moment to talk about coefficient of variation. I think most people have had some experience of the normal curve, bell curve. Right, okay. So we remember that we define a normal curve by a mean, which is kind of the center point, around which it's symmetric. And then around that we have standard deviations away from that. So if we compute the standard deviation of the model, we can ask what is that relative to the mean? If the standard deviation is really big compared to the mean, that's a lot of wiggle room. If the standard deviation is quite small with relation to the mean, we feel rather better about it. So we're looking for those parts of the curve where our our modeled concentration versus the true concentration is low enough in error that we feel the quantitative values have merit. Okay, that's the limit of quantitation. So you can see that this red line that is the coefficient of variation at each of these points that's been estimated out of the model fit. You want that line to dip below some required level of CV, and only the concentrations that fall within, that produce a coefficient of variation lower than that are considered the limits of quantitation. You want the error from the model versus what is observed to be a nice tight fit throughout the LOQ. If you're attempting to quantify outside that, you're guilty of something called extrapolation, about which more soon. Okay, so let us continue on. By the way, I'm including lots of journal articles in the lower bar of this slide. In each case, I've tried to find the paper that does the best job of reviewing this particular phenomenon or illustrating it. So by all means, if I'm making a hash of some of this, feel free to dig up those papers. They may make more sense than I. Okay. We don't frequently use the terms interpolation and extrapolation, but it's actually quite valuable for us to do so. At present, what can we say about the outcomes for people with the COVID-19 disease 1 year from infection? Nothing. Why is that? Hasn't been around a year. It only showed up in December of last year, and we're only in March now. We did make it to March, right? Okay, good. So we're now only about 3 months in. Our knowledge of what happens in COVID-19 disease 1 year out is nil. We have no information upon which to base it. We could extrapolate, right? We could say, well, given that their experience 1 week after cure or 1 month after cure looks like this, we think that a year from now, no symptoms whatsoever. But that is an extrapolation. And we always want to keep ourselves within the data that we actually have. So in the case of the Luminex calibration curves, we always want to be within the limits of quantitation. We want to be able to make our declaration about this fluorescent intensity represents this concentration. We know this because we have concentration standards on either side of it flanking it. So that's an example of interpolation rather than extrapolation. The software understands when it's within or without the limits of quantitation. So in the case of the Luminex platform, you're typically going to see something that looks like OOR less than or OOR greater than. These are out of range. So it is possible that you have measured an intensity below that's too low to be quantified. That's an extrapolation on the low end. It's out of range down. It may be that you have a concentration so great that the intensity is in this blasting zone, basically, for intensity. That creates an out of range high. Now, it could be that this sample really is quantifiable, but it may be that to do so requires a change in the way that you set up the calibration, so that the calibration includes more points in this very low intensity domain or very high intensity domain. It's possible. Sometimes you can deal with the problem in that way. These problems, though, continue percolating forward. It might be that for 50 of your samples, you have a solid concentration for this analyte, and yet when you look at these 3 samples, they got garnished. They're OOR less than. There's so little signal there that it might have been that nothing was present, or it might just be that it was too little for you to detect. Can you tell the difference between those 2 scenarios? You can't. That background light that you're going to get, the base fluorescence is going to mask very, very faint signals. So you can't tell the difference between not present and too little present. All right. So this is generally a problem with cytokines, because when a lot of the times this division is making use of Luminex in the context of cytokines. And cytokines, I gotta say, in proteomics, they're not something that we generally go looking for because they're at such low concentrations. They're hard for us to spot unless we have an antibody to pull them out. Which is why you can do it at Luminex. All right, so that is the first outcome. It might be that your sample needed to be diluted more, right? So if you had a sample that you've loaded 10 times as much as all the other samples on there, the apparent concentration is going to be pretty high, right? So that in that case, you might have a whole bunch of these out of range high values as well. It is possible, and I think that the older you get in academic research, the more apparent it becomes how many mistakes we make every day. I have personally made at least 5 today. So it might seem like you follow the instructions, you get exactly the result you expect, and everything is happy. But the reality is, our hands slip. The calibration on our pipette man might be a little weird that day. The technician you're working with might press the plunger down a little bit more than you do, and so your half of the plate and their half of the plate looks completely different. There are lots of reasons why stuff happens. And those can lead to cases where, in some cases, whole plates need to be redone. And that's really painful because these reagents, they're not cheap. They're not cheap at all. Okay, so, and of course, it may be that in some cases you're working with a clinical specimen that's really, really precious because this is all there is, and you're basically putting the whole sample on there. No dilution, no nothing. And in a case like that, it may just be that there's so little of the sample there that getting an accurate measurement is just not feasible. Okay, so this stuff is going to happen. As a result, we have missingness in our data. You know, if you want to do a t-test and you've got 6 numbers and you're comparing these 3 to these 3, I think most people have some notion what to do. But what happens when you have an out of range up or out of range down hidden among those numbers? Suddenly, figuring out how to plug that, how to plunge that into the calculation becomes rather unapparent. So for that, we have a variety of models that we use to describe what kind of missingness is taking place. We start with a case that we really like, missing completely at random, MCAR. It might seem like this is a really bad thing, but in fact, missingness of this sort is one of the easiest types to deal with. So I want you to imagine that you're an astronomer. And here you are using this fantastically expensive instrument. You have this particular block of time in which to make your measurement. And at the precise moment you hope to visualize a distant galaxy, a plane is flying by. Okay, now, does the plane's appearance over the telescope at that particular moment in time have anything to do with the phenomenon you're observing? Does it matter if the galaxy produces a bright signal or a faint signal? Are they differentially blocked by a plane flying by? No, a plane flying by is perfectly good at blocking the signal from a very bright galaxy or a very dim galaxy. So in this case, we have a missing completely at random scenario. The data are in no way factoring into whether or not you saw them. It's just a chance event. Maybe you're dealing with a microarray or something and you had a bit of lint in there, so this gene probe was not visible. It doesn't matter whether that gene probe is one of great intensity or less intensity. So these are cases where missing completely at random applies. Now, missing at random can be quite a lot more problematic. Now, I want you to imagine that, I want to note that whether the data points are missing or not here has dependence on one of the factors in the study. So let me say that I'm trying to evaluate dieting behavior of full professors, right? And let us say that we measure some men, right? And we measure their weight at the start of the study and their sex and so on. And we measure some women in the study and we measure their weight at the start and the end of the study and so on. Okay, you could plausibly put together something that says, you know, professors, they really need to get out more, because we do. Right. So you can then imagine that some professors, let us say a bunch of old male professors, are actually quite disinclined to share with you their weight because they don't want to get yelled at about it, right? So in a scenario like that, we have a missing at random because some of the professors exhibit this weird behavior and some of them don't. But there's some ability to sort of predict your way around that just the same. So missing at random means that there is a dependence in there, that you can predict which of the folks are least likely to cooperate, in this case based on sex. So you could do that. What's much more frequently the case, and particularly the case in Luminex, is this final case, missing not at random. This is the point where the statistician is going to start frowning at you, missing not at random. In this case, we can predict that the values that are missing in our Luminex analysis are those that have very, very low concentrations and very, very high concentrations. These are a big, big problem. So missing not at random is the worst type of missingness to deal with, and it is exactly what Luminex gives us. All right, so we have some solutions. I'm going to try to walk through what's positive and negative about each of them. List-wise deletion is, well, it's draconian. Does everyone know the word draconian? A draconian solution is one that's very punitive. It's very painful. Imagine that your Luminex kit offers 44 cytokines. Is that a reasonable number, 44? All right, we'll go with it. 44 cytokines. Let us say that 4 of them have a missingness of 50%, which is to say that in 50% of the samples, we had either no data or out of range too low. All right, what can we do with that really? The answer is next to nothing. If you have only 50% of the data points present, frequently you've just got to scratch it. So in this case, we could say these 4 cytokines out of the 44 are simply present at too low a concentration for us to have accurate measurements. Therefore, we are striking them from the experiment. Your design started with 44 cytokines, you're now down to 40. The next thing that could happen is to think about the columns rather than the rows, or vice versa. If instead of looking at cytokines and asking which ones are consistently seen, we instead look at samples and ask, are some samples highly problematic? So let's imagine that I'm doing colon biopsies, something fun like that. I used to work in colon cancer. So it might well be that some of the biopsies were from a nice solid bit that we could clip right off in this person's colonoscopy. Nice solid sample, good hit. But others had very small polyps, and we really had to go sort of scraping the bottom of the barrel, so to speak, in order to get that little bit of sample. In a case like that, it might be that you can get great measurements from the big hunk of meat you clipped out of one person, but this person that had barely nothing to scrape, we're not going to get any decent measurements off of that. So in a case like that, we don't need to be thinking about which cytokines we need to strip out of our analysis. We're thinking about which samples are so problematic they've got to go. Okay? So you can think about this both in terms of which patients need to be stripped out and which cytokines need to be stripped out. But it's draconian. It is a very heavy hammer to lay against your data set. Because somebody who comes to you, like your professor, saying, you know, I was really very interested in, what's a sparse cytokine? Anyone got one for me? No one wants to name a cytokine? Okay, there's a helpful guide to the cytokines on the poster board downstairs in the third floor hallway. So feel free to look at the periodic table of cytokines. It's fine. Well, let us imagine then that some cytokine is present at such vanishingly small concentrations it needed to be stripped out because of list-wise deletion. It is still quite likely that the professor may come to you saying, but I really wanted to know about that one. There is going to be the temptation for you to respond to say, okay, I'll give you the best I've got, and just work with the folks that did have measured values and ignore all the ones that didn't. But what have you done? To do so, just to please your PI, is very dangerous, because now he or she is not going to let go of that number ever, for one, because he or she has just let you know that's their favorite cytokine. But the other thing is that you've thrown out anything that was missing. Only the patients that were missing, and kept anyone who had a reported value. This is not missing completely at random, nor is it missing at random. It's missing not at random. The absent values reflect that you had too little material to get a quantitative measurement. To leave those out means you're losing the low end of your measurements. That's going to bias any result that you pull out of it. Okay, so that's the first solution. It's heavy-handed, but it does work. A lot of people feel that imputation is very simple. All you need to do is figure out what the mean value is for that row of cytokine measurements and simply write it in for any sample in which you had no measurement. Or they say, well, because all of these are missing at the low end, I can simply say what is the lowest recorded concentration that I had for any sample, and I'll write that in for everything. These simple solutions are very bad. So I'm going to show you that a little example here is that before I had a sample of different a sample of different measurements that I'd made, and I had this nice red distribution of values. Then I said, okay, I'm now marking out a bunch of these as missing. I'm now just striking them out of the sample, and I'm going to mark those as missing. Now instead, I'm going to write in the median, the mean value, the average of them all, for each of those missing values. Look how much difference I get in the center of that distribution by imputing the mean. Suddenly I've got this big spike at the mean value, reflecting that I've imputed the mean in those. If you impute the minimum, how does that alter the distribution? Well, now you get this big foothill at the base of your Gaussian, basically. So there are a lot of bad things you can do by imputing a simple value. Either option is going to severely distort the distribution of true measurements. So ideally you will not do this. Someday somebody will ask you to do it. I hope that you are armed to explain why doing it is a bad plan. Okay. Now, I've put Yoda in this figure because he helps me when things are very, very difficult. It's always very helpful for you to have a very smart person nearby who knows how to do complex things. And well-established models for imputing data require some expertise. It's not just anybody in this department you can go to and get this. If you came to me and asked me to do this, I'd be like, oh, no, there's someone smarter to do this. Really. So, in this case, we have some simple imputation strategies, and we have some very advanced imputation strategies. Let's start with simple random imputation. Let us imagine that your missingness is not too bad. Maybe you have 50 people for whom you've measured this particular cytokine, and 48 of them have measured values, but just 2 of them are out of range low. In a case like that, it might be possible, you won't do a huge amount of damage if you are to choose a random measurement of that cytokine and plunk that into each of those 2 spots. So grab for each of these 2 missing places, pick out a random value from that list, plunk it into the first one, pick out a different random value, and plunk that in. That's less damaging than imputing the mean. It's less damaging than imputing the min as well. So this is a start. Most people, I think, could pull off something like that. Make sure you remember which ones you imputed and which ones were real measurements. It's advisable. And Excel doesn't do track changes very well, so it's a good thing to make notes about what you did. Next, a linear model. Now, in the course of your biostatistics unit, you're going to fit a lot of linear models, and it's going to be great. You're going to love it. But I would just note that if you have all these cytokine measurements are not independent of each other. You know, our immune systems work in kind of coordinated ways. And you can frequently find that if you know the set of 43 different cytokines for this person, you can make an educated guess about what value should be there for the 44th cytokine. So a linear model is one of the ways that you can go about doing this. By fitting a model that takes the values of all of the other cytokines to predict this one, you can fill in that value with your best educated guess for it. Finally, you may opt to do multivariate imputation. This is a bit more artful and requires a fair bit of sophistication. And this is definitely one of those situations where I would run to Guillen or to Gerard for their assistance because they're very sharp in this area, which is why I've specified this is where you run to Yoda. I didn't call them that. Okay. All right. Now, there are some sneaky ways to get past this, and occasionally they are appropriate. I have been talking about Luminex as a quantitative technology, where you're trying to say things like, this cytokine is up by 30% in this cohort versus that cohort. Right? That's a quantitative statement. Sometimes, however, we are working instead on a lot of questions about biomarkers. To be able to say, given these cytokine measurements, we want to figure out if this person has data that look more like the case cohort or the control cohort. In this case, you're trying to make a Boolean decision, not make a quantitative statement. Do you see the distinction? Good. Okay, so in a case like that, it is frequently possible to use intensity information directly. In this case, we are not as concerned with translating this into a concentration. We're much more focused on trying to discern if is this value in the high group or the low group. So we almost always have some sort of fluorescence measurement for each cell of our table. Now, some of those we can turn into concentrations, into estimated concentrations. But some of them we can't because they're too low to fall within the LOQ or too high to fall within the LOQ. In a case like that, it is possible to use the intensities directly to be able to say, is this in the high group or the low group? That is a little problematic though, because what if the number of samples you've analyzed was so large that they couldn't all fit on one plate? Are the intensities from this plate directly comparable to the intensities of that plate? Maybe not. So you may need to have some sort of scaling factor that you derive from the calibration curve that was fit to each of those. That might get you to where you need to be. But you're always going to be in trouble if you're relying on cytokines that have such low concentrations that they're almost uniformly out of range low. That's going to be really hard to come back from. All right, now I think that people have a fair number of confusions that they make about about what kinds of data you can use parametric tests on. I think a lot of us know that t-test is useful for discerning whether these two populations come from the same distribution or not. But a lot of us, I think, feel very worried when the data don't look normal. Now, this is a good moment for Gerard to step in and say, the input data don't have to be normal. Their errors versus the model has to be normal. So let me try to explain that point for just a moment. If you were to look at the distribution of heights for all people in this division, what kind of shape would you expect to be? Feel free to just draw in the air. A normal distribution for all people in the division. Does anyone see a problem with this model? What's the suggestion? Okay, so we could imagine a normal distribution where we all have some mean value and we all fit somewhere in that shape. All right. Does anyone see a problem? Well, I don't want to be a sexist, but frequently, men are taller than women. Which means that we don't just have a single normal distribution, we have two normal distributions, and they're superimposed on each other because we're taking the heights of everybody and putting them on top of each other. So effectively, we have kind of this inverted W going on. Now, it's not going to be separated. It's not true that all men are taller than all women. That's junk. And I say this as a man who grew up really short. I swear, when I was 16 years old, I was at my birthday party, and my mom, who was 5'2", minuscule, just put her arm around my shoulder and said, 4'2", his eyes are blue, and I just want to slug her. I hated being short so badly. I'm sorry, I just moved the projector. But I finally started growing. But I've always, in retrospect, thought of myself as a really short dude, right? So when I hit 5'9", I didn't stop thinking of myself as a short dude, right? So when you think about taking the heights of everyone in the department, we have two distributions separated roughly on sex, but they're not perfectly separated, they're quite close together. All right, so if I were to make a model of height in this department, I would want to include as a factor in that the sex, to predict which of these two distributions someone fits into. So when I consider the quality of that model, I'm not going to compare everybody to the group mean. I'm going to compare men to the male mean, and I'm going to compare women's heights to the women's mean. What we care about is that the residuals, which is to say the difference between a person and the corresponding model, is normally distributed. Not that all of that is, that all differences versus the group mean be normally distributed. How does that apply in Luminex? It certainly can, because imagine that cases have a high typical mean and that controls have a low typical mean. What we care about is that the error of the controls versus their mean and the error of the cases versus their mean is normally distributed. So it's kind of a different point, right? You're going to hear that point show up again and again in the Friday morning meetings and stuff like that. So I thought I would explain that one now. All right. So in a case like this, when we're dealing with the data from Luminex measurements, it's tempting to say, well, the data aren't normally distributed. I'm going to have to torture them before I do anything special in statistics. That may not be the case, because you might have a group separation rather than an overall measurement distribution. Okay, so in this case, I have done plots from some real Luminex data collected in this department. And I note that we had some really big problems with out-of-range down measurements. If I look at SIL4R, that's the second one over, I had 36 out of 73 values missing, out of range down. Really hard to come back from that one. For SVEGF-R3, I had 63 of 73 data points missing. Okay, you're not going to do much with that. This is an example of one of those cytokines we just got to strip away, because that's not going to give us any information. All right. So when we look at the data, we might be tempted to say, well, these are all over the map, even for these measurements for which we have a pretty good concentration, as we do in the third case, SIL6R. So how do these data look when we evaluate them as a distribution? Here we're doing histograms. We could do smooth kernel density plots or whatever, but I think the histogram is relatively easy for everybody to understand. So originally, we see that at least half of our data points fall below 10,000 on that plot. We have a fair number that's spread out, that skew off to the right in this long tail, though. And that's kind of worrisome to a lot of folks. It means that we have a very few values that skew to much higher intensities than the others. So one of the things that you'll frequently find is that people say, I shall use log and transform them. Because once you log transform something, it looks normal, right? But I want you to look at what came out of that. When we do the log transformation of those intensities, yeah, it's more symmetric. Those high values have been brought back into the fold, but they're not really, we're not seeing the thin tails that we expect in a normal distribution. A log normalization is not going to fix all of the ills that might possibly occur. What about square root? Now, square root has a nice property in that if you have a 0, it's not going to barf on you. So that's pretty good. If you take the log of 0, you get negative infinity. That's not a helpful number. Right, so you could use square root transformation, but again, not very normal looking at the end. There's a person in this department who really, really likes hyperbolic arcsine transformation. He has his reasons, and you can see that its result is also, well, kind of normal-ish looking, kind of. It has a central tendency at least, a big bin in the middle, so that's something. These technologies are all explained on the Biostat handbook at that transformation webpage, so it's available if you need guidance in doing so. you will frequently hear of our use of Windsorization. Windsorization. Windsorization is very valuable because extreme values are really problematic when you're thinking about central tendencies, when you're thinking about the mean. If you compute the mean for all of the points, the mean gets a little confused when you have a few points that are way high or way low on measurement. So if you'd like to avoid this undue effect, Windsorization is a structured way to alter your data. Now, no one really quivered when I said alter your data. Okay, post-op. Alter your data is a bad thing, right? I mean, you're supposed to use the data. That's why you did the experiment. But in some cases, in cases where you have really high outliers that stand way out from the pack, you need to be cautious about the effect to which those are going to impact any statistic you do. And for that, Windsorization is a structured way to alter the data to reduce this high impact from the outside. So Windsorization means that if you have points that are way out high or way out low, you bring them closer to the pack in general. The rank ordering is the same. The rank ordering. Now, what on earth does that mean? It means that a point that had a lower concentration than everything else before Windsorization will still have a lower concentration than everything else after Windsorization. The highest value before remains the highest value after. But they have been brought closer into the main body of the data. All right. So in this case, the original data had this long tail off to the right. Post-Windsorization, those have been packed right in closer. You can see that the top scale extends above 20,000. After Windsorization, they've been hauled into less than 16,000. So this gives us the ability to avoid the distortion of things like the mean value that would be caused by things being way out on the wing. Several wells of every 96 well plate that you send to Luminex are given over to the establishment of this calibration curve. We need those curves because otherwise we have no way to translate intensities of light into concentrations. Concentration estimates are only reliable if they fall within the limits of quantitation. That's a really important concept for everybody to take away from this lecture. Missingness is a fact of life, unfortunately. I work in the space of proteomics. We're no less likely to suffer from missing values, that you see this peptide has a concentration in these 50 experiments and these 20, well, you just didn't see it. So you have missing values from that. The technologies differ. The problem of missingness is always present, though. Data transforms are very useful for reducing the impact of outliers on the models that we try to create from our data. Luminex assays appear to be a case where extreme intensity highs or extreme intensity lows are a fact of life. And as you will assuredly learn in the statistics module, the greater the variance that you have in these different populations, the less your ability to detect real differences in their means. So being able to reduce this variance in a structured way is actually quite helpful to our modeling from these data.